Today we will talk about the synapse. We first to learn the types of synapses. Synapses are specialized sites for chemical or electronic transmission for communication between neurons and other effector cells, such as skeletal muscle fibers. Most synapses involve chemical neurotransmitters, which are released from presynaptic terminals of one axon or dendrite to affect receptors on the postsynaptic membrane of the target cell. In functional terms, two main types of synapses occur: excitatory and inhibitory. In excitatory synapses, neurotransmitter released from the presynaptic neuron depolarize the postsynaptic membrane. In inhibitory synapses, the postsynaptic membrane is hyperpolarized. Most central nervous system synapses are between an axon of a neuron and the dendrite of another, axodendritic. Other types include axosomatic and, less commonly, axonic synapses. In some sites, such as hypothalamus and posterior pituitary, large vesicles in presynaptic terminals may contain polypeptide hormones. For example, oxytocin and vasopressin, that are neurosecretory products, not neurotransmitters. Then we will see the ultrastructure of synapses. A typical synapse in the central nervous system consists of three major components: presynaptic terminals, synaptic cleft, and postsynaptic membrane. The presynaptic terminal aligns close with the postsynaptic membrane of the target cell. In the area of membrane opposition, presynaptic and postsynaptic membranes are separated by a narrow synaptic cleft, 12 to 30 nanometers wide. Clusters of large numbers of synaptic vesicles in the presynaptic terminal contain neurotransmitter that is released by exocytosis to mediate synaptic transmission. By electron microscopy, synaptic vesicles are 40 to 60 nanometers in diameter and are membrane bound. Whether they have a clear center or an electron-dense core depends on the chemical nature of the neurotransmitter. Pre- and postsynaptic membrane specializations contain electron-dense material that extends into underlying cytoplasm and is usually thick in the postsynaptic area. An action potential causes presynaptic vesicles to fuse with the presynaptic membrane and discharge neurotransmitter into the synaptic cleft. Neurotransmitter then diffuse across the cleft to interact with the receptor molecules on the postsynaptic membrane, which changes postsynaptic membrane conductance. Mitochondria, sacs of smooth endoplasmic reticulum, microtubules, and neurofilaments are also seen in axon terminals. The schematic showing the main features of a central nervous system synapse. We can see the neurotransmitters transport to the presynaptic terminal through microtubules and accumulated, which cause energy. So we can find abundance of mitochondria in the synapse. This is the high magnification of electron micrograph of a typical synapse in the brain. Clear, round synaptic vesicles are abundant in the presynaptic terminal. Some are clustered near the presynaptic membrane in areas called active zones. The postsynaptic membrane exhibits electron densities at two such zones. A narrow synaptic cleft separates the two cell processes. Mitochondria with well-developed cristae are found in both presynaptic and postsynaptic area of the synapse and provide ATP to meet high energy demands. Many therapeutic and recreation drugs with psychoactive effects on the central nervous system have different modes of the action on the synapse. They may mimic a neurotransmitter, block a pre- or postsynaptic membrane receptor ion channel, or affect neurotransmitter breakdown or reuptake. Monoamine oxidase, MAO, a mitochondrial enzyme, normally breaks down the neurotransmitter serotonin. Antidepressant drugs known as MAO inhibitors increase the amount of serotonin available for release into the synaptic cleft. Another class of antidepressants, the selective serotonin reuptake inhibitors, inhibits reuptake of serotonin into presynaptic terminals, thereby increasing its levels available for binding on postsynaptic receptors. Now, that's all for the structure and function of the neuron. So, that's all for the structure and